Today we are re-examining the battle between thalidomide and the human embryo that took place 50 years ago. The result was thalidomide embryopathy. And I shall start with the human malformations, not with the chick embryo or hypothetical molecules. And I shall track the embryonic events by the application of the medical sciences of human anatomy, neurology, radiology, and pathology. Let's first look at the attacker. Thalidomide's pharmacokinetics are critical to this discussion. And what you need to know is that on contact with fluid, thalidomide hydrolyzed into any of 12 products of hydrolysis, none of which was teratogenic. It was the parent molecule that actively damaged the embryo. And here it is in the bloodstream after ingestion at 0 time, reaching a peak in the blood at 1 hour after swallowing and a half-life at 3 hours. This is a stab injury to the embryo. The target was the neural crest, according to my neural crest theory. Now, the neural crest is a primordial tissue, highlighted in yellow in these diagrams. And as development proceeds, neural crest cells multiply, migrate, and sprout axons while they are evolving into the nervous system. Note the diagrammatic axons within the torso of the embryo. Aggregations of the actual crest cell bodies, which contain their nuclei, form the ganglia of sensory peripheral and autonomic nerves. Other derivatives of neural crest include connective tissue of the bones of the face and the skull, the cranial nerve ganglia, conotruncal septum of the heart, odontoblasts of the teeth, melanocytes of pigment cells, meninges wrapping the brain, Schwann cells enclosing the nerves, glial cells, and others. Neural crest is a ubiquitous tissue with many progeny. Now, any hypothesis of thalidomide's teratogenesis must explain the following phenomena. First of all, that the drug action predates the appearance of the limb bud, and secondly, that there is a thalidomide-sensitive period in the human embryo of 21 to 42 days gestation. These facts 1 and 2 were established in the early days of the embryopathy by Lenz in Germany. Thirdly, there was a craniocaudal sequence of occurrence of the defects, established again in Germany by Novak and Lenz. Fourthly, there was symmetry and asymmetry, both seen in the thalidomide series, in all the large published series in the literature. And also in all series, there were upper limb defects more commonly than lower limb defects. Sixthly, there was sparing of the central nervous system. And 7, longitudinal bone loss, triangular bone remnants, dislocations, and dysostoses. There were also associated visceral defects, well described in the British Ministry of Health booklet on thalidomide embryopathy. And finally, neuropathology has been established with the use of thalidomide. This talk is going to examine these 12 phenomena. So first of all, that thalidomide generates upper limb defects before the upper limb bud exists. The timelines of human embryogenesis are very revealing. Fertilization happens here at 0 time. And here we see the sensitive period, which Lenz established to be the time when the human embryo was sensitive to thalidomide as 21 to 42 days of gestation. Lenz's arrow at day 24 shows when the arm defects were first recorded. However, embryology textbooks state that the upper limb bud does not appear until day 28 in the human embryo. So thalidomide cannot act within limb buds because they don't exist for at least another 4 days after its action. Now, molecular hypotheses are located in limb buds. They cannot explain how the drug acts. Not only are the limb buds not present, but the arteries to the limb buds are not present either, so a vascular target is also impossible. But neural crest is present from day 18 and is highly active during the thalidomide-sensitive period, dividing, migrating and sprouting axons. Metabolically active cells are more vulnerable to injury than quiescent cells. This is the basis of many cancer treatments. Cell activity also increases the vulnerability of neural crest cells over other cells in the early embryo. The craniocaudal sequence of events has to be explained. A wave of sensitivity swept down the embryo from head to tail between 21 and 42 days of gestation. Exposure to thalidomide between days 21 and 24 caused eye, ear, cranial nerve and other head and neck defects. Exposure from day 24 of gestation generated arm defects. Exposure from day 27 generated leg defects as well. Novak and Lentz concluded that the morphological type of birth defect was essentially a function of the time of intake of the drug, not the dose. Neubert's laboratory in Berlin found the same craniocaudal sequence of defects in primates exposed to thalidomide. Upper limb defects resulted from exposure to the drug before the upper limb appeared. Neubert et al. Concluded that thalidomide must be acting within the torso of the embryo, deep to the site of the future limb bud. This is where neural crest cells are sprouting axons, and axons are ultra-fine filaments of naked cytoplasm without any myelin protection, and as such they are highly vulnerable. So neural crest is evolving in a craniocaudal sequence that coincides exactly with the dates of thalidomide exposure, and this is circumstantial evidence that thalidomide could be targeting neural crest. Let's deal now with symmetry and asymmetry. The published series of thalidomide cases in the literature reported that the majority of limb defects were bilaterally symmetrical, but they also reported that 15 to 25% could be asymmetrical or unilateral, right to left sides. I asked Professor Nishimura, who had studied thousands of normal human abortuses, whether the right and left sides of the body always develop synchronously, and he told me that they don't. He said 80% develop synchronously, but 20% of normal embryos are out of step, right versus left sides, and therefore develop asynchronously, with up to 4 days difference between the two sides of the body. So it follows that 80% of thalidomide limb defects should be bilateral and symmetrical, and the other 20% should be asymmetrical or even unilateral. Let's look now at the predominance of upper over lower limb defects, which was a characteristic of all the large series. This is a master diagram from Henkel and Willett's landmark paper of 1969, defining the pattern of thalidomide dysmelia in over 200 German proven thalidomide children. The upper limb defects were most numerous, and especially absence of the radius and thumb, this group. Nausea of pregnancy commonly occurs early, at the time of the missed period, and is short-lived. A woman might take only 1 or 2 tablets, sufficient to target the neural crest at about day 24 of gestation, causing arm rather than leg malformations. Now let's look at sparing of the central nervous system. The central nervous system, as many of you know, is the brain and the spinal cord, and the peripheral nervous system lies outside it. The peripheral nervous system has evolved for the signalling and communication necessary throughout the tissues and organs of all animals, and it comprises this huge network of axons extending throughout the body to its very extremities. The peripheral nervous system has been overlooked by some biologists. The basic cell of the sensory section of the peripheral nervous system is the bipolar neuron, shown on the right. In human adults, thalidomide attacks the sensory peripheral neurons, derived from neural crest, not the brain, derived from neural tube. In the embryo, thalidomide appears to target neural crest, not neural tube. The nerve target is therefore consistent throughout life, at all ages. The longitudinal distribution of the skeletal defects was very characteristic. They were called longitudinal reduction deformities of the limbs. Now the skin is innervated in longitudinal strips called dermatomes, each one supplied by a segmental sensory nerve, as shown in this diagram. Here, the dermatome of the sixth cervical nerve is coloured black, and if you excised it and the underlying bones, you will end up with the X-ray shown here of radial aplasia and absent thumb, which was the most common thalidomide malformation. It raises the question of whether thalidomide was attacking the sixth cervical nerve. The skeleton has similar segmental innervation. The strips of segmental sensory nerve supply on the bones are called sclerotomes. Based on past literature and on their own experiments, Inman and Saunders of San Francisco in 1944 drafted maps of the segmental sensory nerve supply of the limb skeleton. When I applied their maps to the radiographs of thalidomide cases, the deformities emerged as failure of formation of sclerotomes. That raises a question. What is the connection between sensory nerves and limb growth? And the answer is that sensory nerves possess an ability to make other cells divide, and this property is called neurotrophism. Neuro for nerves, trophism for growth. Regeneration biologists have proved that limbs will not grow without sensory nerves, whose neurotrophic stimulus initiates mitosis of undifferentiated cells. Nerves recruit mesenchyme cells from the inception of limb formation. Nerves drive cell division thereafter. It follows that if a segmental sensory nerve is damaged, its neurotrophic ability fails, along with all its other abilities, and mitosis of mesenchyme cells will fail downstream in that nerve supply. One or more sclerotomes would therefore drop out of the blueprint of the limb. Developmental biology ignores nerves and neurotrophism and misses the most important factor in thalidomide embryopathy. I took these sclerotome maps to Göttingen in Germany, where Professor Hans Willert and I re-examined his large collection of X-rays of over 200 German thalidomide children to see whether or not the maps of segmental sensory nerve supply could explain the pattern of thalidomide malformations. Compared with the sclerotome maps, the limb defects coincided exactly in 80%, fairly well in 15%, and poorly in 5%. From these strong results, we concluded that both patterns are expressions of the underlying sensory innervation of the skeleton and that the sensory nervous system is involved in limb morphogenesis and limb teratogenesis. Now, there are some other odd features of thalidomide limb defects that must be explained by any hypothesis. These triangular remnants of tibia, femur, and humerus were frequent in all series, and only the sclerotome maps can explain them. First of all, the tibia. Partial tibial aplasia is one of the most curious residual thalidomide remnants. It was a triangular piece of upper tibia bearing on its upper surface the tibial articular surface of the knee joint. Now, the sclerotome maps show that this triangle is the distal end of the third lumbar sclerotome, coloured blue. It lies beside the vulnerable fourth lumbar sclerotome, coloured black, the most commonly deleted sclerotome from the leg in thalidomide cases. Proximal focal femoral dysplasia was common in thalidomide embryopathy, but not confined to it. It occurs in other conditions as well. Loss of the proximal femur, that is, loss of, again, the vulnerable fourth lumbar nerve, leaves an isosceles triangle of distal femur based at the knee. This matches the residual third lumbar nerve supply to the distal femur, coloured blue in the maps. So lumbar nerve 3 resists thalidomide, while lumbar nerve 4 is most vulnerable. Phocomelic arms present variants of this particular complex. The upper humerus has failed to form, and the humerus is reduced to a distal triangle based at the elbow. This is what happens when thalidomide deletes both the fifth and the sixth cervical nerves, blue and black. What remains of the arm falls within the seventh and eighth cervical nerve supplies, brown and green. The humerus is so reduced in length that the forearm and hand move up towards the shoulder, forming phocomelia, and the external shape of the shoulder joint is pointed because of the absence of musculoskeletal tissue of the upper arm and shoulder joint. Dislocations were commonly seen in thalidomide children's joints. And here is the shoulder joint of a thalidomide child on the right, compared with that of a woman with syringomyelia on the left. Both have Charcot's neuropathic joints, characterized by flattening of joint surfaces on both sides of the joint without any loss of bone density, and painless dislocation. Adult Charcot's joints, like the one on the left, occur in long-standing sensory peripheral neuropathy. This can be due to diabetes, syphilis, leprosy, syringomyelia, and other conditions. That neuropathic joints occur in thalidomide embryopathy, as on the right, is evidence that there has been sensory nerve damage in the early embryo. Synostoses, or fusions between bones, was very common in the thalidomide series. And it makes sense if you remember that neurotrophism fails in injured nerves. Mitosis fails in the cells that are going to form the tissue from which the bones will evolve. So the mass of mesenchyme in that tissue is insufficient to complete the formation of separate bones. The result is transverse fusion between paired bones, such as the radius and ulna, the metacarpals, or between the carpals themselves, that is carpal coalition, and or longitudinal fusion between serial bones, that is between humerus and ulna, or between adjacent phalanges. Associated internal defects could form an entire lecture on their own, but we'll have to deal with them briefly. Now, we have seen that neural crest is the source of the autonomic as well as the sensory nervous system. And as you know, the autonomic nervous system supplies all the internal organs of the body with non-voluntary muscles and actions. One assault upon early neural crest could injure both these branches, sensory and autonomic, therefore damaging limbs and internal organs that share the same segmental nerve supply. This indeed occurred. Evidence of such associations is recorded in the British Ministry of Health booklet on thalidomide of 1964. Over 90% of children with congenital heart disease in their records had also upper limb phocomelia of major or moderate degree, indicating major or moderate damage to cervical sympathetic as well as to the peripheral sensory ganglia at the cervical level. Similarly, in that booklet, if you look at the leg reductions, they were associated with anorectal and genitourinary defects, which share a lumbosacral segmental nerve supply. And so, saving the best until last, we come to pathology. Now, first of all, we've got neuropathology in the thalidomide children. There were specimens of bones and joints collected from autopsies and amputations in thalidomide children, and these were examined by Professor Willett and his colleagues in search of histopathology, that is, cell abnormality, the site of the lesion. And although the architecture of the bones and joints was distorted, no abnormal cells could be found in bone or cartilage, indicating that the disease is not in the skeleton itself, but in some other tissue or system, with secondary effects upon the skeletal tissue development. These observations have been confirmed by leading bone pathologists. Let's now look at adults with sensory neuropathy due to thalidomide. In its first epidemic, thalidomide was used as a sedative in adults who complained of tingling, numbness and pain in the feet and hands. German and British neurologists diagnosed sensory peripheral neuropathy of the axonal degenerative type. It was a dying back of axons from their distal tips. 6 years later, Fullerton and O'Sullivan found that 75% of these victims still had symptoms, like this woman shown at the top, whose sural nerve biopsy, 6 years after stopping thalidomide, shows almost complete destruction of her large diameter nerve fibres, that is, the type A fibres that carry touch sensation. The small fibres have proliferated, and that's because it is a fact of neurology that an injured large fibre will regenerate into 2 or more smaller fibres. So her histogram of small fibre sizes is steep and unimodal. The population of large axons has been practically eliminated by thalidomide's neurotoxicity. A normal age-matched control, shown below, shows what a normal sensory nerve should look like at her age. There are plenty of large diameter axons which are normal, and there is a normal bimodal histogram of axon sizes with a small and a large population. Thirdly, we need to look at embryos of rabbits exposed to thalidomide. Rabbits are sensitive to thalidomide. They're a beautiful model, actually. An exposed litter will contain some fetuses with limb defects and some non-deformed littermates. Catherine North compared sciatic nerves of normal controls with 3 groups of thalidomide-exposed fetuses matched for crown rump length. C is for controls, S is for severely deformed, which had absence of the tibia radiologically, M was moderately deformed, who had tibial hypoplasia radiologically, and TND, thalidomide-treated but not deformed littermates, who were radiologically normal in all respects. The results of quantitation of the axons, that is measuring how many of what sizes, showed that numbers of large diameter axons were reduced in all thalidomide-exposed rabbits in proportion to the severity of their leg defects. Even the skeletally normal littermates had neurological damage, proving that nerve damage precedes skeletal damage. Histograms of axon sizes in exposed rabbits, that is, the TND, M and S groups, compared with untreated controls, C, showed a shift to the left towards numerous small diameter axons, even in the non-deformed group. This group represents subclinical neuropathy in the neurologist's terminology. Thalidomide's nerve reduction therefore precedes its skeletal reduction in these quantitative neurological studies, and that's a very important fact to establish. So the neuropathology is the same in the rabbit embryo as in the human adult. Here is a photograph of a 16-day rabbit fetus obtained by Dr. John Cameron. Its mother had had thalidomide, and it shows absence of the thenar eminence. It is not going to be developing a thumb on this side. Dr. John Cameron made transverse sections through the forearm of this rabbit and examined its radial nerve, that is the nerve going to the thumb and forefinger, and here he showed by electron microscopy actual degenerating axons, which he never saw in controls. And again, this is the same neuropathology as in the human adult. So thalidomide consistently destroys sensory nerves. These electron microscope pictures of degenerating fetal axons are the closest we're ever going to get to seeing the actual thalidomide lesion. Neural crest injury explains all the characteristics of thalidomide's teratogenesis. There is consistent neuropathology, and this has been shown in adults of humans, in children, and in rabbits. Thalidomide embryopathy is not a disease of blood vessels, molecules, or genes. It is a toxic injury to immature nerves, leading to disorders of gross anatomy, secondary to demonstrable and consistent primary nerve damage. Within the large lexicon of medical diseases, thalidomide embryopathy belongs to the group of sensory peripheral neuropathies. The pathology is in the neural crest, or the nerve, with the onset of the disease during early formation of the embryonic segmental sensory nerves. Thalidomide, at any age, causes axons to die back from their distal ends, disabling their neurotrophic function in the embryo during the nerve-dependent phase. Mitosis of undifferentiated mesenchymal cells ceases in the band of the injured nerve supply, and a whole population of cells simply never appears. The rest of the limb forms, but it forms minus one or more longitudinal neural segments. To suggest that thalidomide does not attack the neural crest is seen as absurd by informed neurologists, pathologists, paediatric radiologists, paediatricians, and other medical practitioners who realise that neural crest injury explains the embryopathy. The neural crest theory will not go away. It has a life of its own because it explains observed facts. Thank you for listening.